Introduction, and thank you for the opportunity to present. I'm going to talk about collaborative effort here between the J.J. Nuri's lab at the University of Washington and Nadav Aritouf's lab at UCSF. So there's a lot of people involved in actually generating this data. But before I get to show you the data, I want to give a brief motivation. And you're probably familiar with what I'm outlining here. So this is how we envision the precision medicine approach, an approach where we obtain a DNA or an RNA sample of a patient, and then we try to identify genomic variants that hopefully inform us about the treatment or diagnosis of the patient. And while some labs already started doing that, that works maybe to some extent for panels. It works even worse for exomes, and if you get to whole genomes, this is really a challenge. So if you need to sift through whole genome variants, we on the one hand have large collections of annotations that we can use. So we have, let me see whether I can get the pointer over there. Yes. So you can see here, for example, we have conservation information. We have a lot of biochemical readouts from the ENCODE data set. We can measure distance to gene models. So we have a lot of information that we can actually use to interpret our variants. But how to do that? So how to integrate this data is actually quite a bit of a challenge, and several people have or groups have tried to assess though, or to do that, and we have also done that with our tool CAD. But how do you actually test that those approaches actually work? So there was a recent study last year where a group collected known non-coding variants with a disease effect, and you can see these are rather small numbers. So we have a little more than 400 single nucleotide variants where we are rather sure that these are disease-causing. And if you look at those in a little more detail, you can also see that these are not randomly scattered throughout the genome, but rather clustered in a small number of genes. So it is rather hard to assess scores with such a small variant, especially as also a good number of tools is also using these to actually train their tools for variant classification. So rather than using those pathogenic variants, we believe that massively parallel reporter assays, MPRAS, can help us here by looking at a more subtle effect, the effect a variant has on gene expression. So MPRAs allow us to test enhancer and promoters either as a whole element and reading out whether they are driving the expression of a reporter construct or not, or to which extent, better to say, or by testing individual variants, or by what I'm going to present you here, an approach where we're taking one element, we're generating variants on this element, and then we're testing these different haplotypes that have variants in a reporter assay. So what we're doing here is we're using ErrorPro and PCR on a region of up to let's say 600 base pairs in our case, and then we're integrating those haplotypes with different variants into a plasmid that has a reporter gene and a tag in its 3' UTR. And we're going to use this tag, which is going to be associated with a number of variants here, to read out the expression from this plasmid. So we are collecting RNA, and we're also collecting DNA to correct for the number of plasmids that we integrated with a specific tag. In the last year, we've published a protocol that helps us to improve the RNA and DNA readout for those protocols. And in this data set that I'm going to show you here, we've even a little extended that. So we're now using unique molecular identifiers to correct for amplification and other biases in the PCRs. So once we've collected all the counts of RNA and DNA, we can then generate a big matrix of for this tag, how many RNA and DNA counts we have, and with which variants this sequence tag is associated. And we can then fit a linear model where we assign variant effects to each of these individual variants across the region. The data then looks something like that. So you have variants that are repressing below 0 and variants that are activating above 0. And you can already see from such a plot that, as we would also expect, if there's a binding element, the vast majority of effects are going to be repressing. So we can try to start reading binding elements from our data. So we actually looked at a total of 21 elements here with a length range of 187 to 601 base pairs and a rather broad GC content, in total 10 promoters and 11 enhancers. We usually did 3 replicates, so 3 transfection replicates with the same plasmid library. And for 2 of the elements that I'm showing you here, we're also doing complete replicates, so where we are repeating the saturation mutagenesis experiment and we are generating a new plasmid library. And you can see that also among those, the correlations are rather high. So we're looking at 0.92, 0.95 correlation. If we combine our replicates first and compare then you see 0.95 to 0.97 correlations between these experimental replicates. So this is very reproducible, the readouts that we are getting. So as I mentioned, we did a total of 21 elements, and 15 out of those passed our quality thresholds, and they're actually going to be part of the CSKG competition. So there's going to be about 18,000 variants that we have associated with, so where we have a variant associated with an expression effect. And we are asking people to predict these effects. So they're going to get 20% of the data as training and need to predict the other 80%. And while we are really excited to see what people are going to do with this data, we can, of course, already try to take a peek on how available tools perform on this data set. And so I'm showing you here the enhancers, the 5 enhancers that passed our quality thresholds, and the 10 promoters that passed them. And in blue, you're seeing a number of conservation scores, and then in green to reddish, you're seeing the collection of non-coding variant scores. You might not be able to read the axis well. This is something like correlation. It barely achieves 0.5 here. So the predictive power of predicting these expression changes is not immense. To not say for some elements it does not work at all. And you can also see that the order in which the tools predict is not necessarily the same. So there are some tools that are better on certain elements than for others. So I want to show you 2 of the examples in a little more detail. I have to be careful though, because this is going to be data that people want to predict on. So I messed a little with those plots. But yeah, so I wanted to show you that our data also represents some known biology. So these are, for example, the 3 known activating mutations in the TERT promoter. These are frequently observed in cancers. These are the most frequent non-coding mutations in cancer, actually. So this is from a study in 2015 where those mutations have already been studied and where people could show that they are generating new GAP-P binding sites, and that's how they are creating this activating effect on the TERT promoter. We're also finding these other 3 activating mutations here, which also have been previously described in 3 different kinds of cancers. So we think we have a very nice data set here. And again, you can see those activating mutations that we've seen before. And now I'm showing you the whole data set with some modifications. In contrast with conservation scores, functional prediction scores, and 2 panels of motif annotations. So the lower is the motif set that you can obtain from the ensemble regulatory build, and above is a summary of ENCODE annotated motifs. So the first thing that you might recognize is that the known activating mutations are not in the region where we see conservation. So that, I guess, gets back to what we've already heard for protein coding sequence. If something is not conserved, it does not mean that it doesn't have function. So here we're seeing gain of function mutations in a way in regions that are not conserved. Overall, conservation here in this data set doesn't seem very predictive. We see these long stretches which are probably corresponding to binding sites, and you also see them in at least one of the annotation tracks where there's no conservation whatsoever. I already mentioned it now. You have those blocks where we think there should be a motif annotated, but we actually don't have it in the current databases. And we also see these discrepancies between the databases that are available. So if you look at a summary of the correlation here, so these correlations are really low between the functional predictors, which of course integrate this information of motifs and conservation to the actual readout that we get. So I'm going to show you the opposite example now, where the tools seem to work quite a bit better, which is the LDLR promoter. And what you can already notice here, there's again a huge block where we seem to be missing motif annotations. Something that we of course hope will improve also with the new ENCODE data. So I was very excited this morning to see that. But on the other hand, you can also see that something interesting is happening here. So here, those binding blocks that we see in our data, they seem to be, at least on the large level, well recapitulated in the conservation. There are exemptions to that. So there's, for example, this block here, where it's not a repressive region, right? So we learn that conservation does not imply directionality, but we also see this other block where we don't see an effect, at least not in the cell type, which should also remind us that conservation is an organismal effect, and here we are measuring very cell type specific effects. So it might well be that we would be measuring something at this location if we had done it in a different cell type. If we look at correlation here again, we now see that all the tools perform pretty well. It's not amazing, but they're picking up some signal here. So to summarize that, we see that across all these tools that are currently available, conservation seems to be the major driver that they're using for prediction. And however, conservation is not really explaining a lot of our data sets. There are regions where we see very little conservation, and we still see effect. So I'm giving you here a summary across all these 15 elements and the tools that have been, and the average performance of each of the tool. So in our data set, the eigenscores performed the best, followed directly by the vertebrate fast-cons conservation score. And then you see a rather wide range of score performances. What you also probably saw is that our repressive effects, they well aligned with motif data if it was where it was available. Unfortunately, those are frequently incomplete. Something that we are also missing right now in modeling is the creation of motifs, especially those examples that have been the known examples for TERT. We are generating a binding motif for GAP-P. These are, of course, challenges that we need to address in developing tools. So to come to my conclusion, I hope I was able to convince you that saturation mutagenesis data is a very interesting direction to explore, to, on the one side, test our approaches, but on the other also to create trading data sets and to learn from these data sets and identify features that will be interesting to incorporate in new tools. At this point, I have to say that probably the non-coding scores that are available are insufficient to interpret sequence. Yeah, and that we hope that we can develop better scores in the future. So I'm using this opportunity to point out some posters of the group. So there's 2 presentations today. I hope you had the chance to see a presentation of the group yesterday. And I also wanted to point out that we are a very new group and that we are, of course, interested in collaborations and people. So be happy if you contacted us. So then thanks again for the opportunity to present, and I look forward to your questions.
I had 2 questions. So first, I was wondering if you did the same evaluation on this year's KG competition from the MPRAS data from Betty's group?
I haven't, no.
Okay. And then the other question was, do you have a sense of the difference of the performance if you did one of these methods that sort of do the cell type specific predictions? Because I think all the evaluations you had were of sort of cell type agnostic.
Yeah. So currently I'm not testing any tool that is cell type specific. One could include those, and I would love to do that in another analysis. We've only done one of the elements in 2 cell types. So our data is very limited in this respect. So we're going to give the information in which cell type it was done, but we only have one data set where we have the readouts for 2 cell types, and we actually see a change in one binding site coming up. So it's going to be interesting, but yeah.
Hi, Ron Unger, thanks for the interesting talk. It's not a question, I don't know, I would like to hear your opinion. Clearly, there is a lot of variation in expression levels that you have shown. On the other hand, it is clear that there is much more pathological variation within coding regions than based on regulatory elements, right? I mean, everyone sees it and you have shown it. What might be the reason? I mean, maybe the reason might be that, we don't need all of the level of expression, right? Once you reach some kind of threshold, you have enough. It doesn't matter if you change the level further up and down a bit because we don't need it. Yeah. If you change, binding site or even by a little, it will affect everything. Then.
Mm-hmm. Yeah, so I guess we're going to have also 2 competitions for that reason. So one is going to be trying to quantitatively predict the values that we get out, and the other is going to be a yes, no, whether the change is significantly different from 0, so that you get more of a qualitative. So we're hitting a binding site here. I agree with you that there's probably a lot of expression buffering going on, and that the exact level of expression change is not that important. And so I guess my idea of regulatory elements is more that there's a critical amount of elements that needs to come together, and then it happens. And it is, yeah, more about it's a number game rather than, yeah.
Great talk. So have you, how big are these regions? Have you considered, say, the chromatin effect?
Yeah, so the longer regions are 600 base pairs, so they would, in principle,
very small.
Yeah, they're too small.
And they are open chromatin. Everything.
Yes, so especially if you, and I haven't shown that here, if you overlay it with ChIP-seq peaks, right? These are beautiful, totally covered, and they are not informative for that reason. Yeah. The peaks are just too broad for the.
Great, thanks.
Yeah. Okay.
More questions? I have. So it's really interesting to see that conservation is actually not all that it turned out to be, right? So we find that across everything. But I'm wondering if you need to change, to see actually the real effect, to change the alignments, the according to which organism would have this type of tissue and this type of functionality, to sort of filter it by
Okay.
type of conservation.
Okay, that's going to be challenging, but yeah, it is interesting to think about. Definitely,
Do you expect it to have an effect, or?
I mean, people have shown that regulatory elements move around, and I mean, they're short. They're easy to recreate a few bases further on, or another binder can jump in, which would make me a little more hesitant that conservation, even if you did that it would totally rescue it. I would still think that conservation in itself can always only be part of the contribution there. So I think it's an interesting thing to try, but I don't think it fixes the problem.
Well, I'm conservation. I got it. All right.
Okay, thank you.